To wrap up our section on neurodegeneration, I want to turn things to be slightly sunnier and more positive. So far, most of what we have talked about in regards to neurodegenerative disease is about late clinical diagnosis, the devastating symptoms, and unstoppable pathological spread. There are current treatments that slow down the progression of these diseases, and there are a lot of promising looking novel therapies that are getting closer and closer to getting approval. Let's start by talking about what approval means, though. How do treatments actually come to be widely used? In order for novel treatments to become actual treatments, they must go through four phases of clinical trials. If a drug makes it through all stages, the Food and Drug Administration, or the FDA, and doctors will be able to start prescribing it to patients. And these are the four phases of clinical trials. Now, before anything ever makes it to clinical trials, often years to decades of research have been done in model systems, like we talked about in our previous lesson. In order to start testing a novel chemical in people, it will have been thoroughly characterized using in vitro and in vivo models. Once there is sufficient evidence that a compound has beneficial effects, researchers can apply to enter phase one of clinical trials. Now, this first phase is purely to make sure that the drug is tolerable in human studies for the safety of medication or treatment. It's typically just a small number of healthy patients who will volunteer to take this drug for several months to ensure there are no terrible, awful side effects. Sound a little risky? It can be. People are often compensated for volunteering. Once a drug has reached this point, though, researchers are pretty certain that it will be mostly safe for people. And down here, you can see that once a drug enters this phase, it has a 70% success rate, meaning 70% of the compounds that enter this phase will make it through into this next phase. This next phase, phase two, is the really big one. This phase is when a drug will be tested in patients affected by the disease. People with this disease will either be giving a placebo or the drug and have their symptoms very closely monitored for a minimum of 6 months. If the patients that receive the treatment show statistically significant improvements or a slowed progression, this drug will progress into phase three. Looking down here, though, this phase only has a 33% success rate because often these drugs simply don't help enough with the symptoms. For the drugs that do make it into the next phase, this is the long one, phase three, where dosing and efficacy are really started to be detailed, which involves many more participants and lasts for 1 to 4 years. This gives clinicians a much better idea of the extent to which the drug could be beneficial in the long term and how widely applicable it could be. But here we see a 25 to 30% success rate of drugs that enter that will continue on into the last phase. So phase four is the cherry on top to really finalize those very important details like cost effectiveness. And I say that sarcastically. But by the time a drug reaches this point, it's pretty likely to reach full approval. You'll see down here, this has a 70 to 90% success rate of actually continuing on into full FDA approval. If you do this math though, with all of these percentages, only about 6% of drugs that enter phase one will make it all the way to FDA approval. And I want you to keep that in the back of your mind as we talk about current treatments, as well as treatments that are going through this process. That being said, the current treatments for neurodegenerative diseases are not great right now, even though they've gone through this long vetted process. The majority are designed and used to alleviate symptoms, but at the moment, there's nothing that stops or slows pathological progression. So let's take a look at the treatments and how they treat the symptoms. So looking at Alzheimer's disease, let's focus on memory loss portion and treatments that exist to alleviate memory loss. So researchers first had to determine a target for memory, which led to the cholinergic hypothesis of Alzheimer's disease. Acetylcholine is an excitatory neurotransmitter that contributes to long-term synaptic remodeling. Specifically, it's well known that high acetylcholine input leads to long-term potentiation, or LTP. Now remember, LTP is following this main idea that cells that fire together will wire together. So cells that communicate often are able to communicate faster and easier. Conversely, low acetylcholine input is known to lead to long-term depression, or LTD, where communication between cells becomes slower and more difficult. Acetylcholine is known to interact with pathways that heavily influence both tau and APP. If we look here, we see how decreasing acetylcholine affects both phosphorylation of tau, such that more tau is phosphorylated, and also changes the way in which APP is cleaved, leading to increased amyloid beta and decreased glutamate signaling. If we think about this glutamate signaling and circle back, pretending like this axon is synapsing on a new neuron, we have decreased glutamate signaling, meaning we'll see long-term depression between cells. So, in order to try to increase acetylcholine treatments, researchers have targeted acetylcholinesterase here, which is the enzyme that degrades acetylcholine, leading to the class of drugs known as cholinesterase inhibitors. These are used to treat early to mid-stage Alzheimer's. And by using cholinesterase inhibitors, more acetylcholine stays around longer, presumably rescuing this long-term potentiation by not having this increased phosphorylation and not having this increased amyloid beta. And we do see, to some extent, that cholinesterase inhibitors slow memory decline. This is an older study from 2001, which reported the effects of 6 different cholinesterase inhibitors in patients. And all of these showed that they were successful in slowing symptom progression. So in this graph, we're looking at change in cognitive score, which means an increase is worsening memory. So when we see out here that they don't continue to change in their score, that's a good thing. However, cholinesterase inhibitors have some serious side effects. Acetylcholine is the primary neurotransmitter that exists at the neuromuscular junction. So you can imagine that, in addition to some gastrointestinal issues, other side effects include things like cramps and falling, and some other central nervous system effects like confusion and hallucinations. And even more frightening, these can lead to death by heart attack, as acetylcholine is essential for proper pumping of your heart. As patients progress into later stages of the disease, cholinesterase inhibitors become less helpful. And I mentioned in the previous slide that glutamate was something that was heavily affected in Alzheimer's disease, which brings us to the glutamate hypothesis of Alzheimer's. You hopefully recognize this as the primary excitatory neurotransmitter of the central nervous system. And in late stages of Alzheimer's, some research suggests that dysregulation of the glutamate system leads to glutamate being randomly dumped into synapses, which can lead to excitotoxicity, or cells dying from being overstimulated. To prevent this, memantine is commonly prescribed. It is an NMDA receptor antagonist, meaning it blocks glutamate from binding to NMDA receptors and thereby prevents neuronal death by excitotoxicity. Now that was just touching on only the memory loss of all of these symptoms we see here. That is not even an exhaustive list. So what about some of the other symptoms, such as changes in mood? These symptoms are commonly treated again by targeting the symptoms and repurposing drugs used for other disorders and diseases. For example, for depression or anxiety, patients will often be prescribed antidepressants, such as SSRIs. Or for psychotic episodes and paranoia, antipsychotics will very often be prescribed, typically treatments that are used for schizophrenia and bipolar disorder, such as clozapine and haloperidol. Again, I want to emphasize that these are treating the symptoms, and these are drugs that weren't even made specifically to treat Alzheimer's disease. These are drugs that were made to treat other diseases that have similar symptoms. So let's switch over and take a look at Parkinson's disease treatments. Most treatments here have focused on the hallmark motor symptoms, like the tremor and the bradykinesia. And we know that these symptoms are caused by dopamine dysfunction, specifically by degradation of dopamine cells in the substantia nigra, which interrupts typical signaling in the nigrostriatal pathway, which affects the dorsal striatum, thalamus, and hypothalamus. This knowledge led to attempts to replace dopamine. Levodopa, or L-Dopa, was first used in patients in the 1960s and is still the most commonly prescribed treatment for Parkinson's disease. L-Dopa is a precursor to catecholamines. And just as a reminder, catecholamines are a way to group neurotransmitters, which includes dopamine, as well as other ones like norepinephrine and epinephrine. L-Dopa can be taken orally as it has the ability to cross the blood-brain barrier. Now, though, it is commonly supplemented with this other drug, carbidopa, which prevents L-Dopa from being degraded out here in the periphery, because we want L-Dopa to cross the blood-brain barrier and enter the central nervous system. In order to further help, there has been additional drugs that is leading to this cocktail treatment that also help get L-Dopa into the central nervous system and help turn it into dopamine and help it stay dopamine. So this is a complicated schematic that I threw up really quickly, but I want to emphasize this here. We're looking at L-Dopa, being able to cross the blood-brain barrier, thanks in part to COMT inhibitors, preventing it from being degraded in the periphery. Once it's in the central nervous system, COMT inhibitors prevent it from going this way and promote it to actually be turned into dopamine. When we have MAOB inhibitors, this prevents dopamine from being metabolized into DOPAC, which is one of its inactive metabolites. So we're getting drugs to get L-Dopa into the brain, turning into dopamine and staying dopamine. And L-Dopa does improve symptoms. It reduces overall scores on a Parkinson's rating scale and specifically reduces the motor impairments that are seen using this particular Parkinson's rating scale. However, of course, this treatment is increasing dopamine throughout the entire brain. And the nigrostriatal pathway is not the only dopamine pathway. This is an untargeted general sledgehammer type of treatment. And increasing dopamine in this way is going to affect other brain regions and other dopamine pathways, like the mesolimbic pathway, which sends dopamine signals from the ventral tegmental area to the nucleus accumbens. And you may recognize these regions because these make up the reward-seeking pathway. So when you know that, it makes sense that increasing signaling here leads to side effects of increased reward-seeking and decreased inhibition. A major side effect of taking L-Dopa is gambling and pursuit of sexual urges. And it's not uncommon for patients to gamble away their life savings or become very promiscuous. And I don't think I really need to highlight how destructive these behaviors can be. So patients are really carefully watched, especially in the first few months until their dosing regimen is figured out. Increasing dopamine can also lead to dyskinesia or increased uncontrollable movements. Remember that basal ganglia circuit? We're trying to stimulate it with more dopamine. And sometimes this leads to overshooting a little bit and can worsen a patient's tremor. So, overall, this dopamine replacement therapy approach is a sledgehammer approach and it works, but it's not the best. Same as with Alzheimer's, the other symptoms of Parkinson's disease are treated with repurposing of other drugs. Taking just a really quick look at Huntington's disease, there are very, very few treatments. Tetrabenazine is the only current FDA approved drug specifically for Huntington's disease. And this decreases monoamine neurotransmitters, so doing exactly the opposite of what L-DOPA is doing. And the idea then is to decrease the signaling going on in the basal ganglia circuit. And overall, that's not great. For the non-motor symptoms, again, we're receiving repurposed drugs. And I'll have to say that all of these diseases, people are just being asked to take a cocktail of pills every day to manage symptoms while their diseases continue to worsen. So I promised we'd be a little more positive. So let's see where we are now, where we're getting this big, big push to focus on the pathology rather than the symptoms. And at the beginning of this entire section, I said we'd be looking at distinctions and similarities of neurodegeneration. And one major similarity that these diseases are characterized by is aggregation of specific proteins. And this led researchers to refresh themselves on some basic philosophical logic. I'm married to a philosophy major, so I couldn't help but bring in a logical argument. So if we have our first premise that misfolded protein X causes disease X, our second premise that if we have more of protein X, it's more likely that protein X will aggregate, we can conclude then that if we decrease protein X, we can slow or stop the formation of new aggregates. This means that researchers are focusing their efforts on decreasing protein levels using techniques known as RNA interference and antisense oligonucleotides, or ASOs. Both of these work in similar ways. The main goal is to stop translation of RNA into protein. Let's revisit the central dogma of biology, the translation from RNA to protein. So the DNA is the architect of our body. It gets transcribed into RNA, which then leaves the nucleus. And we can think of RNA as, construction managers. In a normal system, these construction managers will translate the architect's wishes into proteins. And then we can think of proteins as the contractors. They are the ones that build and maintain the cells in our body. ASOs, for example, interrupt this system by swiping in and binding to the RNA, which will prevent translation into the protein. RNAi or RNA interference works in a really similar way by targeting the RNA at this point, usually by cutting up the RNA into small, unreadable pieces. These are both really great because they can be designed to be highly specific and target very specific known sequences. For example, alpha-synuclein and Huntington. And people have been really excited about the potential of RNAis and ASOs to actually address the underlying neuropathology, truly stopping the progression of these diseases. A lot of effort is being put into developing these drugs, which is clearly portrayed here. And this is just showing most of the ASOs that were in clinical trials for different neurodegenerative diseases and which stages they were in and this is now 4 years old. Since this came out, this graphic, the ones up here for spinal muscular atrophy, Huntington's, and ALS are still ongoing. However, this APP one down here has been terminated during the second phase, as it was clear that patients were not improving with the ASOs. Not on here, though, is an ASO called B2B094, which is currently in phase 1 for Parkinson's disease. So there are a lot more that are still, up and coming, hoping to enter these clinical trials. And all of this is still very early. But one thing that I like to keep in mind is every single day we are closer than we ever have been to finding a cure for these diseases. And the only way we'll continue to get closer is if we keep doing research and keep working to better understand these diseases. If you're especially curious about the most up-to-date information and new treatments, you can use this link to actually search ongoing clinical trials at the National Institute of Health. And I highly recommend you go take a look around.